Well, thank you so much for inviting me to speak today and for that kind introduction. These are going to be the lesions I'm going to talk about today. I have no relevant disclosures. We'll start off talking about tumors in the retina and then move on to the optic nerve. And to start with, I just wanted to review retinal histology and the glia you find in the retina, since some folks here may not look commonly at eyes. So in the retina, you have radial glia, which are known as molar glia, and like other radial glia, they're going to span the entire extent of the retina. So you have the cell body here in the actually not working. The cell body in the internuclear layer and these long processes that extend to the internal limiting membrane and the inner aspect of the retina and out to the photoreceptors. Interesting, these molar glia only become GFAP positive in a reactive state. The normal resting molar glia aren't GFAP positive. As with other radial glia and elsewhere in the brain, these have a lot of stem and progenitor properties and can become quite proliferative. Astrocytes in the retina are largely restricted to the nerve fiber layer. This is the axons from the ganglion cells that are going back to form the optic nerve. You can see them as these elongated oval cells in the nerve fiber layer. When you stain with GFAP, you can see these around blood vessels and with the extending processes in the nerve fiber layer down into the interplexiform layer. Maybe a hint of radial glial processes in the deeper portions of this retina as well. As in the brain, these astrocytes are important in forming a barrier with blood vessels, and here on this GFAP stain you can see the GFAP positive astrocytic processes abutting the blood vessels extending down into the retina. It's sort of interesting, these glia in the retina, they enter the retina relatively late in fetal development, and they sort of spill out of the optic nerve head and move externally towards the retinal periphery and act as a guide for the growing vasculature. So they're really quite intimately associated with the overall development of the retina, and particularly with the development of the retinal vasculature. So turning to our first tumor today, this goes by a number of different names, probably most commonly called retinal astrocytic hamartoma, sometimes called giant cell astrocytoma of retina. These tend to occur in patients who have a tuberous sclerosis. Occasionally you can see sporadic lesions or similar appearing tumors in patients with NF1 or retinitis pigmentosa, but generally associated with TS. They tend to be rather benign or indolently growing lesions. Clinically, they have two main types of presentations. They can be sort of smooth and non-calcified or more heavily calcified and sometimes multinodular. Here's an example of the two major microscopic appearances of the tumor. They can be comprised of sort of bland spindled cells, and other lesions have more of what's called the giant cell component, where you have in the lower right, where you have these larger cells, prominent nuclei, big nucleoli. Now, for the neuropathologists in the crowd, and you look at these giant cells down on the right, this will of course remind you of another tumor that we see in the brain in patients with tuberous sclerosis, which is the subependymal giant cell astrocytoma. And really these astrocytic hamartomas in TS patients in the retina basically have a spectrum of appearances which is quite similar to what you see in SIGAs in the brain. Here's an example from Wilmer. This one, you can see this very discrete tumor arising from the retina in fundoscopic exam. This one happened to be highly calcified. You can see all these refractile spherules of calcium sort of popping out of the tumor on the left. Over here, use this as a pointer? No, yeah. So over here on the right, notice how sharp the demarcation is between the more normal looking retina and this heavily calcified tumor mass. Then you can see here some of these larger cells that are somewhat SIGA-like in the middle of this calcification. Here's another example from our lab of a well-defined tumor. You can see here very sharp lateral edges arising from the inner retina in the posterior pole. Here's the optic nerve coming in. at higher magnification, you can really see how it's not invading into retina in any significant way. The retinal morphology on either side of this particular case is well maintained, and you just have this sort of bland spindled mass of glial cells, some limited blood vessels in there, but it's not heavily vascularized like the next type of tumors we'll talk about. The higher mag view, again, showing these very bland spindled cells, glial cells, lack of mitotic activity. Now, there have been some nice studies correlating the pathology in patients with tuberous sclerosis with other clinical features. This was a series from Cleveland Clinic in which they looked at 132 patients with TS, and in those 132 patients, 36% had these astrocytic hamartomas in their eyes, and in 43% of the patients who had tumors, they were bilateral. So as with retinoblastoma, they can be bilateral. And in patients who had these retinal lesions, they more frequently found clinical findings such as cognitive impairment, epilepsy, and SIGAs in the CNS. So manifesting these in the retina is going to be associated with more aggressive features elsewhere in these patients as well. Occasionally, these tumors can behave in a more aggressive fashion. Here's a series of 4 cases that was published by Dr. Eagle. You can see these are larger tumors than the ones I'd shown you previously, and here in this PO, or this rather eye in which the initial collat's been taken off, you can see a detached retina here, this whitish substance, and then a very large tumor here adjacent to the detached retina. These are more pictures from Ralph's paper. Here's another case, really a huge tumor filling the eye, neovascularization on the surface of the iris due to hypoxic tumor, similar to what we just saw in retinoblastoma. Here's one, a cut through the optic nerve head, and you can see not just the bland cells I'd shown you in those cases from Wilmer a second ago, but in addition, some necrotic areas, but the viable cells did still have this sort of SIGA-like morphology. Again, another parallel to SIGAs, this is still pictures from Ralph's paper, is the fact that these are really, while we're calling them astrocytomas, they express both glial and neuronal markers. So these large giant cells have essentially a biphenotypic expression of glial markers such as GFAP and neuronal markers, and in this case, NSE. Ralph had noted that some of the giant cells in his tumors express neuronal but not glial markers. Now, for these more aggressive tumors, obviously it would be nice to have a chemotherapy that could be used rather than enucleation. And so with SIGAs, of course, there has been some success in treating them using drugs that's going to target the pathway that's activated, such as everolimus. And so here's data from a published study in which about half of the patients showed a 50% greater reduction or more greater reduction in the size of these brain tumors after 2 years of therapy. There's not as much data from using similar approaches to treat tumors of the retina, but there was a paper that came out about a year ago now from China in which they had looked at 7 patients, and there were multiple lesions in the patients. And so here on the right, they show the percent decrease in size of the tumors that were being treated with sirolimus. So you're getting between marginal decreases to maybe a 25% decrease in tumor size. But I think there's real promise here in using these sorts of therapies for aggressive astrocytic hamartomas in tuberous sclerosis patients. So what other tumors, what other gliomas can you see in the eye? Well, really probably the most common one that you might encounter if you look at eyeballs at your institution is the so-called vasoproliferative tumor, also known as massive retinal gliosis. As we'll discuss, neither of these names, which are the most commonly used ones, are perfect, and I think there's some new proposals that may be better. And these generally arise in eyes in which there's some inciting event. It could be a sort of chronic inflammation, uveitis, trauma, congenital malformations, retinal detachment. There's something that gets the ball rolling in these cases. And they're basically comprised of tightly packed spindled astrocytes and are generally associated with prominent vasculature. Here's an example from our institution, a 75-year-old male in which the eye was enucleated 20 years after there had been a ocular trauma, and you can see this white mass of glia filling the eye with numerous fairly large vessels scattered throughout the lesion. When you look under the microscope, you can see these variably sized vessels with blood, and then all these densely packed solid masses of proliferating tumor cells in between the blood vessels. Just a higher magnification view, and you can see how there tends to be a perivascular orientation in these spindled glia around the blood vessels. It's somewhat similar to what you can see in ependymoma, and I'll discuss in a second the possibility that the suggestion some have made that these might be ependymoma in the eye. So, as I mentioned, the nomenclature is variable, and I think both of the historical terms have some issues with them. The term vasoproliferative tumor sort of implies that this is primarily a vascular problem, but I think most people believe now that the vascular elements are secondary, and in fact they're not always present. So, really it's a problem of glia. Thus, I think this name is not optimal. Massive retinal gliosis, which goes back over 50 years now, is a well-accepted term. It does capture a lot of what's going on. However, in a significant number of cases, including the initial case series in which this name was coined, it's more of a focal nodule. It's not a massive gliosis filling the eye. So there's some problems with this terminology as well. So here's, for example, a case from Wilmer, where it was an eye that was enucleated from a 33-year-old man who had chronic inflammation known as uveitis and retinal detachment, and over 3 years this thing grew. But you can see, so here's the sclera, here's the lesion. This is a relatively small, you don't have a sense of scale, but this is a relatively small focal thing, again with the prominent vessels in it. So this is not massive, really, in the grand scheme of things. Here's histologic pictures from this same tumor. Again, you can see a fairly sharp demarcation from adjacent retina. Here's the lesion. Unlike the astrocytic hamartomas of TS, again, you have all these large blood vessels in this particular case, which you can see on higher magnification here, and another high mag photo here. It looks very similar to the prior case I showed you. Occasionally, and it was true in this case, you can have these more elongated processes that in this case are not really around blood vessels but are near the underlying choroid. So it has been suggested that maybe these are ependymomas in the eye. This is really the main paper putting that view forward. Bernd Scheitauer was a proponent of this notion. There wasn't a lot of genetic data or clinical data. It was more based on morphology, and there were some EMA dot-like inclusions and microlumens in there that would be similar to what one sees in ependymoma. I would say this notion really hasn't gained traction in the community at this point. My personal thoughts would be that unless you could show some sort of genetic similarity, overall, I don't think these are ependymomas. I think the perivascular orientation is more just the way these glia grow in the eye. So there was another study that came out recently addressing some of these issues that include a number of neuropathologists, Ari Perry, Dan Bratt, as well as the ophthalmic pathologist at Emory, Hans Grossner-Klaus, who is the senior author. And they addressed they looked at 4 cases, and they were asking, the question, one, is it really the astrocytes of the blood vessels that are primary drivers here? They felt like this is mostly an astrocytic proliferation with secondary vascular involvement. They noted that they can see sometimes Rosenthal fibers in eosinogranular bodies in their tumors. I tend, in the 10 or so cases I've seen, I've not seen so much in the way of Rosenthal fibers or eosinogranular bodies. I don't think those are common features of these lesions. And they started to get at the important issue of is this a reactive or a clonal neoplastic lesion? And they looked specifically for molecular changes found in gliomas in the CNS, including BRAF fusions and IDH mutations, and they didn't see those in those 4 cases. Hans had proposed the name reactive retinal astrocytic tumor for these because he felt like that encompassed, you better characterize what was going on. And I tend to agree with him. I think this is probably a superior nomenclature. There's only limited additional genetic data for these. Here was a study of a case report from Japan in which they'd used a Humira androgen receptor assay and had not found any clonal, presumptively neoplastic event in this a single additional case. There's not been a lot of gene expression profiling. Here's a very recent study from Deepak Edward, an ophthalmic pathologist practicing in Saudi Arabia, and they had done expression microarrays in the tumor from this 28-year-old gentleman that had grown over 18 months. And basically, the major pathways are upregulated were not surprisingly astrocytic markers, inflammatory markers, extracellular matrix markers. There's been suggestion over the years that these tumors arise from molar glia, but in this single case, they didn't find any signs of upregulation of molar glial markers, suggesting that maybe it's not originating from these radial glia, but from astrocytes. And he also had not seen a lot of upregulation of vascular markers in this single case looked at by gene expression arrays. Now, we've looked at a couple cases that have come in the last year or two using molecular analyses. Here is one that was an in-house case, sort of a standard-looking case, masses of GFAP positive glia, large PAS positive blood vessels, and we had sent this for array CGH, and it was there were no karyotypic abnormalities by array CGH in this case. But there was a second case we had gotten in consultation from Sarah Coupland, who'd sent it from England, and Ralph Eagle had looked at it as well. Her case was a very aggressive one. Unfortunately, I couldn't find the photos of it. But it actually filled the eye and perforated through a posterior scleral defect. So it was growing very aggressively in the eye, and although the morphology was fairly typical of these lesions. And interestingly, when I sent that case for array CGH, there actually was monosomy 6. So in some of these, you can see genetic alterations, and it's possible that some of these actually do have clonal genetic changes in them. But really, I think we just don't know a lot about what's going on genetically in these tumors. Finally, I should mention that if you start reading the literature, you will see a handful of case reports. They tend to be older, claiming to see oligodendrogliomas in the eye. The nerve fibers inside the eye in normal eyes should not be myelinated. There's not oligodendroglia there. So it would be surprising to find oligodendrogliomas in the eye. And in fact, these case reports are largely just based on H&E morphology, where they said, wow, there's lots of little round cells, and I think it must be an oligodendroglioma. There's no genetics backing this up. Some of these have subsequently been shown to be retinocytomas of the sort that Ralph had shown in the last talk. So, but just be aware that there are papers out there claiming that there are oligodendrogliomas inside the eye, but I don't think there's a lot of good data to support that. So let's now turn to the subject of gliomas in the optic nerve. So just to, again, orient folks to the optic nerve, I think most of you are well aware of this, but for non-pathologists in the crowd, as you get closer to the eye, you have the central retinal artery and vein, the arachnoid and dural coverings of the nerve, and then all these packets of axons coursing back to the brain. And at higher magnification, you can see that here, there are lots of oligodendroglial cells, but also lots of astrocytes in these cells that are separated by PL septa. Here's just a picture of a GFAP stain out towards the lepto meninges, and you can see the GFAP negative lepto meninges and all these astrocytes in the optic nerve. So almost all of these optic nerve gliomas, of course, are pilocytic astrocytomas. Occasionally, very rarely, you'll see gangliogliomas and high-grade astrocytomas in the nerve. There was a series of 1,200 orbital lesions reported from Wills by the Shields, and 4% of them, 48 of them, were optic gliomas. That gives you an idea of how common they are as orbital lesions. About a quarter of patients have evidence of NF1 who have optic gliomas, and of course, in the context of NF1, these can be bilateral tumors. Here's a case which nicely highlights the typical growth pattern in the nerve. When they're in the optic nerve proper, they tend to sort of expand and distend the dura like a sausage, which you can see radiographically. And then when you take these specimens out, you have this very distended sausage-like growth of the optic nerve. They only get outside the dura very, very, very rarely. It's like a case report to see these things get outside the dura of the optic nerve. In this particular case, you see it extending posteriorly back towards the chiasm. And of course, as we'll see later, these kind of rise first in the hypothalamic region or chiasm and then extend down the nerve. When you look at these gross specimens, sometimes the tumor stays mainly within the parenchyma of the optic nerve proper, but often you'll see it spilling out into the arachnoid, the subarachnoid space. And there's been a bit of controversy and perhaps some ongoing controversy over the years as to what this proliferation in the leptomeninges is. And particularly in older reports, they refer to this as meningothelial proliferation. And indeed, that there is, you certainly can see true meningothelial proliferation out here. But in the 50 or 100 true optic nerve gliomas that I've looked at, I would say more frequently it's actually glial tumor cells proliferating out here in the coverings and distending. So you can see both, but I think more often this is going to end up being glial tumor than leptomeningeal cells. Here's a cross-section, a picture I took from one of Peter's books. And in this particular case, there was a tumor in the optic nerve proper, but also a lot of glial tumor out here in the leptomeninges. So of course, the morphology is similar to what we see in pilocytic astrocytomas at other sites. One can find, generally speaking, Rosenthal fibers in the tumors. One can find eosinophilic granular bodies in the tumors. But you can also see other variants occurring in the optic pathway gliomas, particularly those rising in the hypothalamic region around the chiasm can be of the so-called pilomyxoid variant. So, and I'll show pictures of those. Oops. Oh, it's not. I'm sorry, this is the wrong version of my talk. Okay. I'd inserted some nice pictures last night of pilomyxoid astrocytomas, which I guess we reverted somehow to the earlier talk. Anyway, so the pilomyxoid astrocytomas, of course, have a blander morphology. You tend to see these bland spindled cells, often with myxoid stroma surrounding blood vessels. And they're quite common in younger children in the hypothalamic region. For a time, it was thought that they might represent a unique entity. However, with time, we've come to realize that the genetics are quite similar to the broader spectrum of pilocytic astrocytoma. And in fact, you can have tumors not infrequently that present initially as a pilomyxoid astrocytoma, and then following treatment, they'll recur. And when you look at the histology of the recurrent tumor, it's matured and taken on a more classic pilocytic appearance with Rosenthal fibers, dense pyloid regions. In other cases, you can have lesions that present initially at initial resection with some areas that look pilomyxoid, other areas that look more classically pilocytic. So at this point, the thinking is that pilomyxoid change is not reflective of a distinct entity, but just part of the spectrum, maybe an immature, more aggressively growing spectrum of what you can see in pilocytic astrocytoma. So what is the natural history of pilocytic astrocytoma? Well, most of them remain stable. However, some can spontaneously regress. So while a lot of them are stable and spontaneously regress, there is a significant subset that will grow and impact vision and cause blindness. In terms of the syndromic cases, it tends to be the sporadic non-syndromic cases that arise earlier and are more likely to grow rapidly. The NF-associated ones tend to be a bit more indolent. The genetics is been worked out pretty well over the last 5 to 10 years. We're still learning new things. But the most common genetic event are changes in the BRAF oncogene. And a number of labs around the world, about 10 years ago or so, were looking at pilocytic astrocytomas. This is just a paper from our group. And we all found sort of the same thing, which was duplications of a 2 megabase region that encompassed the BRAF locus. And what that duplication causes is a fusion between the active kinase domain of BRAF and the promoter region and N-terminal coding region of a KIAA gene. And it's the RAS binding region that's not included from BRAF. So this part of the BRAF gene that's not included in the fusion is the negative regulatory region that keeps the kinase activity in check. So what you end up with is diffuse expression in neural tissues of this constitutively active BRAF kinase. And so this is quite a common change you can see in these. And we had tried to model this in human neural stem cells. We took human neural stem cells and introduced, initially shown here, the BRAF V600E. Subsequently, folks have used the fusions and see similar things. And what happens is when you introduce these active BRAF kinases, there's an initial proliferative burst and clonogenic burst of growth. But then eventually those cells are going to stop growing, and that's very similar to what you see in nevi on your skin, where you have solar damage to the melanocytes. The melanocytes proliferate, they form a mole or a nevus, but then by and large that's going to stop growing. And that process is thought to be mediated by oncogene-induced senescence, where the same BRAF kinase eventually is going to induce P16 and P53 that are going to shut down proliferation and make things stop growing. Now occasionally in the skin, of course, they don't stop growing, they progress to melanoma, and that's generally in the skin associated with loss of P16, loss of the brakes or checkpoints that are the drivers of senescence. And we can see similar kinds of P16 loss in the more aggressive pilocytic astrocytomas. So when you look at pilocytic astrocytomas, at least in our study, we found that P16 loss at the protein level is associated with a more aggressive behavior of these tumors. And Nada Habato's group, at the same time, we had published this paper 5 or 6 years ago in Clinical Cancer Research, had published a paper where she showed in culture also the loss of P16 is important in terms of allowing these tumors to grow through oncogene-induced senescence. So finally, this slide came out as well. Another slide I put in was last night was showing earlier studies from Fausto Rodriguez when he was still at the Mayo Clinic, in which he had shown by FISH that in anaplastic pilocytic astrocytomas, there is where you have a high mitotic rate, necrosis, things like that, so-called anaplastic features, a high percentage of those have biallelic deletion of P16 as well. So occasionally, as I mentioned, these things will start growing in the hypothalamus and secondarily extend down into the optic nerve. So here's an example of one of these that presented initially in a 2-year-old. And this tumor has some pilomyxoid features. You're not seeing the dense pyloid gliosis Rosenthal's. This maybe not the perivascular orientation, but some pilomyxoid features to this one. And this tumor grew progressively through chemotherapy and radiation all the way down the optic nerve out to the globe. And when there's some distension and loss of architecture to all these individual bundles of axons because there's tumor cells growing in here and making them expand. And when you look under the microscope, you can see little Rosenthal fibers. We found EGBs as these tumor cells are growing down these nerve fascicles. And eventually, it made it all the way up to the lamina cribrosa. It did not grow into the eye. So this particular case respected that boundary and didn't grow into the eye. This did have a BRAF gene fusion and showed P16 loss in the tumor. So we wanted to look at pilocytic astrocytomas of the optic nerve proper. A lot of these early studies we did were optic pathway or other sites. And so we had worked with Doug Cameron, who was at the AFIP at the time, and found 59 patients from the older files. So the reason we have to go back to the older files, of course, is now we no longer are going to surgically excise or even treat some of these optic nerve pilocytics. So you really have to go back 30, 40, 50, 60 years to find cases in which the eyes and the optic nerves were enucleated in large numbers. And so we were able to find 59 of these in the AFIP files. And we were able to do then some genetic analyses on them. So another thing that came out, basically only 15 of these, so we had 59, only 15 of them could we successfully do FISH, looking for BRAF duplications, but 11 of the 15 where we could successfully do FISH had the BRAF duplication. So a very high percentage of these in the optic nerve proper had the BRAF duplication. 4 of the cases were from NF patients, and interestingly, there was one of those NF patients also had a BRAF duplication. So you can occasionally see both NF and BRAF duplications in the same patient in these optic nerve pilocytics. So this is data from a review article we'd written, and so now there's a larger number of cases here because we're now, I just given you numbers, those 11 of 15 were just for tumors that arose in the optic nerve proper. If you expand your thinking to all of the optic pathway, going back to the hypothalamic and chiasm, and looking in the published literature and our own series, you can get to a larger denominator. Using that, about half of them are going to have the KIAA BRAF fusions, which is a number similar to what you see in brainstem or spinal cord, but more common than you would see in cortex or deep gray matter. Of course, the most common place you're going to find these alterations are in the posterior fossa. So there are some treatment implications for this. When it was first discovered that BRAF is a major driver, people got excited about drugging this pathway, and there was an early study of treating progressive, aggressive pilocytics, which was headed by Matthias Karianis here at NYU using sorafenib. Our institution was also involved in this trial. 13 children were enrolled, but we saw a paradoxical increase in growth in those cases, similar to what you can see with this paradoxical activation of signaling in the skin sometimes. And so that trial was shut down early because of the paradoxical increased growth that was seen with sorafenib treatment. There's been a lot more success in targeting this half of things, which you might not think would be active in these BRAF-driven lesions, but in fact they are. Here's some data from our primary optic nerve glioma series where Fausto Rodriguez had stained for a number of markers of mTOR pathway activity, and you can see in these primary optic nerve gliomas, there's a fairly high percentage of them that are expressing immunohistological markers of pathway activity. This is probably the largest trial to date. It's closed. These data are from a published abstract from SNO a few years ago. This is a trial headed up by Mark Kieran at Dana-Farber, and they had enrolled 23 patients and treated them with RAD-001, and they'd shown that they were able to hit targets in these optic pathway. Well, these weren't just optic pathway. This is pilocytics at multiple sites. They were able to hit targets. 4 of the kids had a greater than 50% reduction in tumor size, 13 had stable disease, 6 had progressive disease. So there's some promise there, and there's some new trials that are coming that are combining everolimus with other kinds of therapy. Here are 2 children from that trial that were treated at Johns Hopkins, and you can see some of the more successful cases. Here's a case in which you had a very large hypothalamic lesion, and it didn't shrink in size, but it sort of hollowed out. You sort of lost all the central contents of the tumor, although there wasn't a lot of clinical progression in this case. Here's another case where it was much more dramatic, where you had a large lesion that essentially totally regressed on therapy, although it came back subsequently when they were through therapy. So I think clearly this by itself is not going to be sufficient, but it's probably going to be a successful component of some sort of multimodal therapy for these more aggressive pilocytics moving forward. I've looked at, as was stated in the introduction, I live the Notch pathway, and we've done some studies looking at Notch that turned us towards optic pathway gliomas. This was a paper we published a number of years ago in Cancer Research, in which we had introduced constitutively active forms of Notch3 into the growing fetal eyes and brains of mice. And we got these, this is a small one, but we got very large ones that grew out of the optic nerve into the orbit. But this is like a baby one of these tumors, very bland, sort of oligoid glial cells growing underneath the leptomeninges. So this is the optic nerve of the mouse. This is the retina of the mouse. But these could dramatically distend and overtake the whole optic nerve of these animals, but retain a somewhat bland cytology similar to that you would see in a pilocytic. So that would suggest that maybe Notch can drive tumors in mice, at least, that look like pilocytics of the optic nerve. And in fact, when we looked at a number of human pilocytic astrocytomas, including 4 or 5 of these were lesions from the optic pathways, and looked at a number of Notch pathway components, either by RT-PCR at the mRNA level as shown on the left, or immunohistochemically as shown on the right. We could see that the Notch pathway was significantly upregulated. There's normal brain here on the far right. So there was significant upregulation of Notch pathway receptors and targets in human pilocytic astrocytomas. We also did some broader gene expression profiling studies. Shown here in red on the left is normal brain, and in green here on the right are a number of different pilocytic astrocytoma cases. And again, you see upregulation of Notch pathway markers in the pilocytic astrocytomas. So there is significant upregulation at a broader mRNA expression profiling as well. And finally, we've done a little bit of work. There's not good pilocytic astrocytoma cell lines that are out there to work with. This is a pediatric low-grade glioma cell line, but not a pilocytic. And when we genetically silence Notch signaling using hairpins targeting the canonical transcriptional cofactor, it slows the growth of these. So some hope that maybe Notch inhibitors might work in these lesions as well. Just as a side note, for those of you trying to grow pilocytic astrocytomas in culture, or as xenografts, it's just really hard. They'll grow for a few passages, but basically all of your tumor cells are going to rapidly senesce. And if you can actually keep things growing. So I've tried to grow like 20 of these over the last 15 years as xenografts, neurospheres, high serum adherent cell cultures. We're using conditioned reprogrammed cell conditions right now. And in all of these, you can get things to grow and you get excited, but then when you use FISH or other markers and try to prove that the signature genetic change in the primary tumor is there in what you're growing in culture, you don't find it. And what you're actually growing is contaminating astrocytes. And the actually genetically defined pilocytic component is going to be gone rapidly. So I would encourage any of you trying to make models of these. If you think you've made a model, prove it to yourself on the genetic level. And I'm sorry to say, if you're ever not disappointed and actually do grow something, I would love to talk to you and use that in a collaborative fashion. So you occasionally can see other tumors in the optic pathways. Here's a case I got from Fausto of a ganglioglioma that arose in the optic nerve. A lot of it was glial, but there were some ganglion-type cells in the tumor, and the synaptophysin and chromogranin stains were highlighting this ganglion cell component. So this isn't a great one, but we convinced ourselves this is a real ganglion cell component. You can see them occasionally. Very uncommon to find high-grade astrocytomas of the optic nerve. For the neuropathologists, obviously, just as true at other sites, pilocytics can have lots of vascular proliferation. They can be fairly cellular. You can see some mitotic activity. As long as you believe that that's a pilocytic astrocytoma, those things are not going to make this a high-grade glioma. It's still just a pilocytic astrocytoma. As I mentioned earlier, you can have anaplastic pilocytics, but they're all pilocytics. Occasionally, you see true infiltrative gliomas in the optic nerve. I've only seen 3 or 4 of them, I think, maybe over the last 20 years. But I wanted to share a case that we had just last month. It was really spectacular. So this was a 42-year-old woman who had presented to the ophthalmologist with visual loss in her right eye, had edema of her optic disc. They did an ultrasound. They found a dome-shaped lesion overlying her right optic disc that was about 3 millimeters thick, and saw some enlargement of the optic nerve behind it on ultrasound. On imaging, you can also see the thickening and a lesion growing in the right optic nerve. And so the history of this patient is important because she had been diagnosed with an anaplastic astrocytoma of the brain 5 years previously that had been treated initially with surgery and Temodar, had eventually progressed to GBM. This was an IDH1, known to be an IDH1 mutant tumor that had lost ATRX and had been treated with Avastin and a PD-1 inhibitor, but neither of those things were working very well. And so here's a picture of the initial tumor in this patient, or rather the MRI of the radiographic appearance of the original tumor back in the frontal lobe. Here's what it looked like under the microscope, malignant-looking astrocytoma, clear vascular proliferation, clear necrosis, so clearly a GBM after it had progressed. And so they went in and did a vitrectomy where they stick a needle back into the back of the eye and suck out some vitreous, and they also tried to get a little bit of the superficial tumor. It wasn't really an FNA per se, but they can sort of grab small bits of tumor off the surface of this mass in the back of the eye. And you had these clusters of sort of hyperchromatic cells. There weren't great glial processes, although we didn't have smears to look at. In other areas, the cells look higher grade, and there were some apoptotic or necrotic cells admixed with this sort of wispy stuff is the vitreous collagen up here on the right, somewhat fairly discohesive thing. And these cells were strongly GFAP positive. A subset of them expressed the glial marker OLIG2 in their nucleus. They were strongly p53 positive, as was the primary tumor. They had a high proliferation index on KIAA1549, and generally we don't see rapidly proliferating normal things from inside the retina. And we could see expression of mutant IDH protein on immunohistochemistry and loss of ATRX in the neoplastic cells with some rare stromal cells that seem to show retained ATRX. So I think in this case, what you had was this GBM grew up the optic nerve and into the eye. So, but this obviously is not something that you're going to see every day. But in this case, I think the studies strongly suggest that we have a genetically identical tumor that's grown into the eye that had started back in the frontal lobes of this patient. So I'd just like to stop with that case and thank the folks in my lab who've contributed to the research studies I showed you over the years, as well as our collaborators and funders. Thank you very much.